Good morning, everyone. My name is Gerold Schmitt-Ulms, and I'm a researcher at the University of Toronto. And I'm going to talk to you today about a prion protein lowering treatment approach that we're pursuing in my laboratory. As you know very well, PrP lowering approaches are very promising and are a validated strategy for extending survival in prion diseases. This particular approach started a few years ago when a team member, Declan Williams, discovered that the prion protein, shown here in yellow, often sits in the brain right next to another protein, which is a pump molecule, a so-called sodium-potassium pump. This molecule uses energy to intrude potassium atoms while it's extruding sodium atoms. And this molecule is really important because these atoms are actually charged and they produce the electricity that we use in our brains to think and to do a lot of things that are important for cognitive function. So cells have developed all sorts of tricks to make sure that these pump molecules are always present at the cell surface. The prion protein is less important for cells. What we realized once we understood this relationship, that they are sitting next to one another, we formulated the hypothesis that we could perhaps exploit this to get rid of the prion protein. Now, how would we do this? A lot of people have tried to find small molecules that can bind to the prion protein itself. But this molecule is a bit like Teflon. It doesn't really bind anything very well. But these pump molecules, they have a channel. And there is actually natural compounds known as cardiac glycosides that can intrude into this channel and can block it, thereby inhibit the pump. When this happens, because the cells so much rely on these pump molecules, they will immediately try to replenish the inhibited pump molecule. The way it does that is the cells will internalize the pump molecule and degrade it, and then send a new fresh pump molecule to the cell surface. And what we were hoping was that the prion protein sitting next to the pump would get co-internalized and co-degraded. And this is exactly what we later found to be true, as I'll show you in a moment. But first we had to solve one other problem. These molecules are known as cardiac glycosides, and the word cardiac indicates to you that they are actually affecting the heart. And this is very true. They're not known to pass into the brain very well, and that's where we need them to be to treat prion disease. So at that time, we teamed up with this expert for cardiac glycosides, Pavel Nagorny, who works in Ann Arbor, Michigan. He is the world eminent expert, in fact, on synthesizing cardiac glycosides from scratch, from very basic building blocks. He can do so in less steps than anyone else. And we recruited his opinion on how we might turn these cardiac glycosides into molecules that can get into the brain. And he suggested that we can change certain atoms here on these molecules, shown in red, green, and blue R labels. And we could stick various other groups in these respective positions. So we tried this in silico, meaning computationally initially, predicting the potency and predicting how these changes might change brain access. And in the end, we settled with one particular molecule, which had a high predicted potency and very good brain access, better than any other one. Oops. And this molecule is known as now, or we termed it KDC203. And here is now a type of data that you might never have seen, but I'll explain very briefly to you how it works. It's not complicated. Here's 5 samples. Each sample is a lane that you see here with the numbers 1 through 5. And when we don't treat with KDC203, we see a black blob here, and that is basically the signal that indicates the amount of the prion protein that's present in cells that we have processed to get these signals. And here you can see as we increase the concentration of KDC203, the intensity of the signals disappear for the prion protein. So we are in fact reducing the levels of the prion protein dramatically, more than 85% when we quantified this. These first data were in human cells that we kept in culture, but we can also do this with brain in a dish cultures. What this is we take a rodent brain, we slice it into slim slices to maintain the architecture that the brain cells have. And then we treat them also with KDC203 levels here indicated as we increase the concentration. You see here that the prion protein level in an untreated sample had a particular intensity, but then it gets diminished. Next, once we had established and validated that this protein can be this molecule can be potent, KDC203, we tested in a small mammal whether it actually gets into the brain and has the effect also in a living mammal. And I must say, these data were utterly disappointing. What we observed was that several of the animals had strongly reduced levels, but they were all over the place. And we had blinded ourselves to which animal was getting which concentration of KDC203. When we unblinded the results, we learned that there was absolutely no correlation. And it caused us a lot of head scratching. And at this point, we understand that as we had given these compounds orally, and as they were traversing the gut, bacteria in the gut actually derivatized or changed the chemical composition of KDC203. So it no longer was KDC203, and it then didn't have the potency, and it didn't get into the brain in the way in which we had hoped it would. So our possible solutions for this would be twofold. We're again working with Pavel Nagorny on this at the moment. One is we can create a stealth KDC203 related molecule that would be resistant to this inadvertent conversion that occurred in the gut. Alternatively, we could look into whether we can administer KDC203 or a related molecule not orally, but through a blood-based infusion. We prefer if we could keep the oral route, because then it would be really easy. One could give a patient once a day a tablet, and that would be good enough to keep that level of PrP reduction as we like it. There is a few characteristics of cardiac glycosides that keep us hopeful that this is indeed a really good strategy going forward. One is this class of compounds is actually very well understood. There is 250 years of pharmacological data that humans have collected. So the clinical experts all understand cardiac glycosides really well. And at the concentrations at which these compounds are PrPC lowering, they're not toxic at those concentrations. But perhaps one of the strongest arguments for not giving up on this strategy is that in the end, if we were to succeed with a molecule that has that stealth characteristic, can be given orally, it would cost about $1 a day to treat a patient. When you compare this to some of the other strategies that are being pursued at the moment, they would likely cost, if current cost levels are can be used as a comparator, they would likely cost 100 to 1,000 times more. So let me conclude by thanking Debbie Jobs and the many good people around her to make this work happen. But also I realized that none of this would be possible without the dedication and generosity of many of you in this room. And I would like to highlight these families that are listed here on this slide. Thank you so much.